though it's 4 degrees Celsius outside. So today we have part 3 in our 3-part series on the nervous system. Just making sure I have all my here. Okay, so we started talking about the nervous system with kind of big scale organization. What are the parts of the nervous system? How does it work kind of at a macro level? Then last time we talked about action potentials. We talked about signals within a single cell, how you get that signal from one end of the cell to the other. Today we're going to talk about how you get that signal into the next cell in line. So we're talking today about synapses. And then if we get there, we're going to talk about sensory receptors. I will tell you, yesterday, we ended up talking for just like 20 minutes about how memory and learning works in here, and we're going to bring it back to sensory receptors, and that's okay. So we'll see how far we get today, and we'll get as far as we get, but we're not going to rush. So we have 4 parts today. We're going to start with synapses. We're going to sequence an HD, synaptic transmission. So we're going to look at the structure of synapses, the different components, and then see how they actually work. Then, once we establish that, we'll talk about memory and learning, because memory and learning is all about your synapses. So compare and contrast, short-term and long-term memory, and also sequence in HD, the process of long-term potentiation. A lot of the stuff in here is actually going to be a review from the first class of Gen Bio all the way back in the fall, but we're going to add some new detail now that we've actually talked about the nervous system a bit. Then comes our second half. We're going to talk about sensory processes. So part 3, we're going to look very generally at how sensory processes work. What are the steps in sensory reception? What are the components of those steps? So sequence in HD, that pathway. And we'll finish by looking specifically at human vision. We rotate through a bunch of different senses year after year. Last year we did ears. This year we're going to do eyes. And I know it's none of you in this room because the people that come to class on Friday morning, I know you're all with it. But for your colleagues who may be less diligent, who are using previous year's notes, just let them know that we haven't done eyes since before the pandemic. So, like, if you've got old notes, they don't have eyes, unless they're exceptionally old notes. So you can either tell everybody that, or you can keep it to yourselves. I'll leave it to you. But I know everybody in this room is diligently taking new notes for this year, and it won't be a problem. So let's talk about synapses. It would help if I plug this in, I think. Help if I plug it into the USB port there. Yeah, there we go. Someone who's been doing this for a while might think to plug their stuff in. All right. So last time, as I said, we talked about action potentials, and the big thing is 1, how an action potential happens, and then 2, how you get that action potential from one end of the cell to the other end of the cell. Today, we're going to talk about how one neuron, having sent an action potential down its axon, is going to transmit that signal to the next neuron in line. That's the big thing that we want to start with here today. Those junctions, of course, are called synapses. So let's just very briefly, in a big, broad way, introduce the concept of a synapse, and then we'll zoom, as we like to do, we'll zoom further and further in. We'll see how it works at the molecular level. Okay, so a synapse is a junction between a synaptic terminal and another cell. So here we have, highlighted in the red box, we have a synapse between 2 neurons right there. So this is what we call a presynaptic neuron, and this one is the postsynaptic neuron. And there's going to be a signal that goes down this axon, it's going to get across that synapse into the next cell in line. Now for today, we're going to talk about this in the context of one neuron signaling another neuron. But that doesn't have to be the case. Think about the pathway we talked about last time. The afferent side of the peripheral nervous system, the central nervous system, then the efferent side, and it ends with an effector, like a gland or a muscle. synapse there, and we're going to talk about that actually next time when we talk about how muscles work. We'll see synapses between motor neurons and muscle cells. So it's going to be structurally a little bit different from what we're talking about today, but they'll work basically the same way. So the specific context we're dealing with today is one neuron signaling another neuron, but you can have other types of synapses where it's a neuron signaling like a muscle, for example. It all kind of works the same way, and we're just going to stick within the nervous system today, but it's all doing the same basic thing. So there are two types of synapses. One of them is an electrical synapse. And this is the only slide we're going to have on electrical synapses today. I just want to introduce the idea of it. And we're going to see a little bit more of this when we talk about cardiac muscle and how the heart works. So we'll see this a little bit next time, and then when we talk in a couple of weeks about the circulatory system, we'll come back to not exactly electrical synapses, but a structure within cardiac muscle that works basically the same way. The idea with an electrical synapse is rather than have an action potential in one cell and then an intracellular chemical signal trigger an action potential in the next cell, an electrical synapse is a direct connection between the cytoplasm of two adjacent cells. And it's very similar conceptually. The proteins involved are different, but it's analogous functionally to plasmodesmata that we talked about in plants earlier in the semester. So this figure does a pretty good job of showing how this works. You have one cytoplasm down here. You have a little ring of proteins that makes a channel through the plasma membrane to both cells. So if you have a depolarization of this cell, if you have an action potential right here, those sodium ions can diffuse directly into and depolarize the next cell, triggering an action potential there without needing an extracellular transmission step involving neurotransmitters. Okay, so that's kind of structure is called an electrical synapse. And we're not going to worry about that for the rest of today, but again, we will see a kind of similar structure when we talk about cardiac muscle in a few places, a little bit later this semester. We are going to focus on chemical synapses today. These are the big ones. When you think synapse, what you're generally thinking about is a chemical synapse. These are the ones that involve neurotransmitters. And these make up most of the synapses in your body. It's mostly going to be chemical synapses. So we're going to use this figure to do two things. We're going to look at the structures involved here, what are the different components. And then we're going to go through the process. We're going to start with this cell right here. We're going to talk about what happens at the gap, and then we're going to see what happens in the next cell. So we're going to start with this. And then once we get to the receptors, we're going to zoom into that postsynaptic cell as we go through the process. Okay, so here's our main components. We have a presynaptic cell. That is the cell that is sending the signal. So this red arrow indicates an action potential. So you're going to get an action potential down through the axon of the presynaptic cell, and that's the signal that we're going to jump into the next cell. You have to get across a small gap. It's called the synaptic cleft, and this is an extremely small gap. If you think back a few weeks to when we talked about during Endocrine 1, when we talked about the different types of signaling. One thing we did when we compared all those different types of signaling, we talked about how some of them are long-distance signaling, some of them are short-distance signaling. This is the shortest of short-distance signaling. I'm measuring this in nanometers. Okay, so this is about as short distance as you can get. And that's important because how are those neurotransmitters getting from one cell to the other? It's just diffusion. So the shorter the distance, the faster and more efficient that diffusion is going to be. And think about if you can perceive a lag. If you go to write something, you move your arm, right? It happens when you want it to happen. There's no lag there. The reason for that is because this is really, really fast. And so triggering the action potential, we're talking milliseconds. Getting across that gap is milliseconds. And the reason for that is because it is such a short gap. If it was longer distance, it would take longer. But we don't really, when you're doing endocrine regulation, especially for lipid-soluble hormones, it can take its time. When you're doing synaptic transmission from your nervous system, you better do that right away. So we make sure that this is a very small gap. And then on the far side of that synaptic cleft, we have the postsynaptic cell. That is the cell that is going to receive that signal and respond to it in some way. And the main thing that we're going to emphasize today in terms of a response is changing the membrane potential of that cell, either depolarizing it or hyperpolarizing it, and potentially triggering a new action potential in that postsynaptic cell. So those are the parts. What questions do you have now before we get into the actual process of a chemical synapse? All good? All right. So we're going to start in our presynaptic neuron up here. And in fact, we're going to start all the way up in the cell body. Because in order for synaptic transmission to work, you need to have neurotransmitters. And if you think back to last time, the structure of a neuron, all of the stuff you need to make things, like proteins, neurotransmitters, vacuoles, all that stuff is way up in the cell body. It's not in the axon. So you have your nucleus, your ER, your Golgi, your ribosomes, that's all way up in the top of that cell there. So you make your neurotransmitters up there, you package them into vesicles, and then those vesicles or vacuoles, whatever you want to call them, structurally they're the same. They're going to be transported down that axon, and the way that happens is really cool. You don't need to know this. If you take like a cell bio class, you'll learn this. But the way that works is it actually has we have transport proteins in our cells, and those transport proteins move along the cytoskeleton like it's like a commuter rail line. So you have things moving throughout your cells from end to end along the cytoskeleton. And in a neuron, that means they hop on all the way up in the cell body, and then they basically walk down the cytoskeleton, down the whole length of the axon, dragging these vesicles with them that are filled with neurotransmitters. So that's how the vesicles full of neurotransmitters get from where they're made in the cell body all the way down to the axon terminal down here. So we make our neurotransmitters, we load them up into synaptic vesicles, and then we transport them all the way down to the terminal. And now we're ready, right? We have a bunch of vesicles that are ready to go. But we don't just want to release them any old time. We need to be told to release them. We want to very specifically, very tightly regulate that process. So the next thing that's going to happen is you have action potential, right? Red arrow, action potential. It's going to depolarize this cell. And when we get down to the terminal, we have a new type of voltage-gated channel that's going to play a role here. We've previously talked about sodium voltage-gated channels, potassium voltage-gated channels. At the synaptic terminals, we have calcium voltage-gated channels. So those calcium channels open, and that causes calcium ions to flow inwards. We didn't talk about this last time when we talked about membrane potential, but your cells also establish a calcium gradient, where there's more calcium outside your cells compared to inside. So if you open up these voltage-gated calcium channels during an action potential, that's going to allow calcium to flood into that synaptic terminal. Now, have we seen anything recently involving calcium doing stuff, like a wave of calcium through a cell? Have we seen that happen recently in any context? Where was that? Where did that happen? Fertilization. Now, when we talked about fertilization, three things happened. Did any of them involve vesicles that were in the immediate vicinity of the plasma membrane, that calcium wave? Cortical reaction, right? Remember what happened when we did fertilization? You have the calcium wave. In that case, it's released from the ER rather than from outside the cell, but it's the same effect. You get a wave of calcium going through that cell, and in that at the time secondary oocyte, you had those cortical granules that were arranged around the underside of the membrane, and that calcium wave caused those vesicles to fuse with the membrane and release the things they contained. In that case, it was enzymes. They release them via exocytosis. The exact same thing happens here. It's exactly the same thing, except the vesicles that fuse with the membrane and release their contents, they don't contain enzymes, they contain neurotransmitters. But it's exactly the same process. The calcium rushes in, that causes the synaptic vesicles that are immediately adjacent to the membrane to fuse with that membrane and release their neurotransmitters via exocytosis. It's exactly the same process as the cortical reaction. My fingers are like, it is so cold in here, and I have cold air blowing directly on them. This is, it was 2 degrees Celsius when we woke up this morning. The air conditioning is on. So this is one of the critical steps here, right? We've released the neurotransmitters. Is everybody okay up to that point? And the neurotransmitters are released, we're ready to go. So the neurotransmitters are going to get into that synaptic cleft. And 3 things can happen there. We only care about one. I don't care about the other 2. I don't care if you know the other 2. So some of those neurotransmitters, even though it's really short distance, some of them just like diffuse away and they get lost, whatever. Some of them, they ping back and forth, and they end up getting taken up by the presynaptic cells, called reuptake. But we don't care about that either. The most important thing that happens, and where most of those neurotransmitters go, is they're going to bind to the receptors on the postsynaptic membrane. That's what we care about, so that's what we're going to pick up with on the next slide. Okay, so we've got our up to the postsynaptic receptors. Everybody okay with that? Ready to go? Yeah.
How does the rush of calcium cause the vesicles to...
How does it cause it to fuse? I don't know. I do not know. And there are 2 options, and I'm not sure which is right. So one option is that it's about the charge, because calcium has 2 units of positive charge, and things like sodium and potassium only have 1. So maybe it's something with that. Or the other option is that there's something specific with calcium, where the phospholipids on the outside of the vesicles and the inside surface of the membrane, there's something specific about the way they interact with calcium. It's not just that it's a positive charge with 2 units of charge. It's specific to that ion that causes them to fuse together. I don't know what the answer is, but good question. So that's, if you want to start keeping track, that's the first question today that I didn't know the answer to. I think we may have hit 10 yesterday. Yeah. Just a little bit louder, please. If the calcium doesn't enter, no neurotransmitters get released. Yes. Yep. Which is a really good example of why it's really important to maintain homeostasis for like a bunch of different things, right? If you have a calcium out of balance thing, it's not just like a bone and osmoregulation thing. It's also going to mess with synaptic transmission. Just a lot of important things here. Okay, so our neurotransmitters diffuse across that synaptic cleft. They're going to bind to postsynaptic receptors. A really important thing with these postsynaptic receptors, a theme that we have seen before, is that they are specific. We are not going to talk today about any specific neurotransmitters, nor any specific receptors for them. There's a picture I'm going to show you later on. It's got a lot of details in it, and the receptors are labeled. But we're not going to worry about the labels. One type of receptor is purple, the other is pink. I'm just going to call them purple and pink. It doesn't matter what the receptors are. Right? Next time, when we talk about muscles, there is going to be one type of neurotransmitter I need you to know. Anyone know what the neurotransmitter is you're going to need to know for next time? Just say it out loud. Just, yeah, acetylcholine. Good. So we'll do that next time. Of all the that's the only specific neurotransmitter you need to know in this class, and it's not relevant for today. But conceptually, for today, a really important thing is that the receptors are specific to each neurotransmitter. So you're going to have receptors for this or that neurotransmitter that are only responsive to that specific signal. Okay, that's the big important thing here. And we've seen that before with other types of signaling, right? We talked about how important this is with endocrine signaling, that you have receptors for each specific hormone that don't react with the wrong thing. That's going to disrupt homeostasis, and obviously the same is true here. So you need to be specific in terms of detecting that signal. So now let's talk about the effects in the postsynaptic cell. The result of synaptic transmission, when the postsynaptic cell detects those neurotransmitters, is that we are going to generate a postsynaptic potential. And that is just a change in the membrane potential of a postsynaptic cell. Because remember, the ultimate point here, if you want to send a signal through multiple cells, is you have an action potential and then a synapse. and then you need to trigger an action potential in the next cell. In order to do that, you need to change the membrane potential of that cell so that it hits the threshold and triggers an action potential. You accomplish this through the use of ligand-gated ion channels. And the term ligand is just something that binds, right? So something binds to something else. The thing that binds to the other thing is usually the smaller thing in the pair. We call it the ligand. So in this case, the neurotransmitter, which is this like red guitar pick thing right there, that's a neurotransmitter. That's the ligand. It's going to bind to its receptor. And the receptor functions as an ion channel. But it's a new type of channel. It's not a leaky channel. It's not a voltage-gated channel. It's not one of those that we've talked about already. It's a new thing. It's a ligand-gated channel. So when that neurotransmitter binds to that protein, it opens the channel and allows, for example, sodium ions to diffuse through. And when that happens, then it's going to depolarize the membrane and potentially trigger an action potential in the postsynaptic cell. That's the big idea of how this works. Use the neurotransmitters to change, depolarize that cell so that you get another action potential. You could also go the other way. You could hyperpolarize that cell, because maybe this isn't a sodium channel, maybe it's a chlorine channel. Right? Chlorine is a negative ion. So if chlorine floods into the cell, that makes the membrane potential greater, makes it less likely to trigger an action potential. So you can also inhibit transmission this way. It just depends on what ion can get through that channel. And just like it's specific for the neurotransmitter, it's also going to be specific for the ion. So we can go either way. Yes? That is facilitated diffusion. Yes, it is. Excellent. Good. Now, there's two types of postsynaptic potentials. These names are long and complicated, but they basically just mean depolarization or hyperpolarization, like we talked about last time. So an excitatory postsynaptic potential, or EPSP, that just means you depolarize that target cell, which brings it closer to the threshold to trigger an action potential. Right? It makes it easier to continue that signaling pathway, so we call that excitatory. And the opposite is an inhibitory postsynaptic potential, or IPSP. That's going to hyperpolarize. If you hyperpolarize, you get further from the threshold potential, so it's harder to generate an action potential. So an IPSP is going to suppress or inhibit an action potential in that next cell. This graph shows the effects. There's a problem with this graph that I'll get to in just a minute. So we have on the y-axis is membrane potential, our baseline here at -70 in yellow. And then the green bits are the parts where you have an excitatory postsynaptic potential. So it depolarizes the cell, brings it closer to that threshold. The red parts are inhibitory postsynaptic potentials. They hyperpolarize, so they make the difference greater and make it harder to trigger an action potential. And as you can see, if you have enough excitatory signals, then you hit the threshold and you trigger an action potential, like we talked about last time. But the problem with this graph is nothing to do with the content. The content is all good. The problem is that we're using green and red to show opposite things. That is bad graphic design. Never do that. Why not? Because green and red is the most common form of color blindness. So there's a very good chance somebody in this room is looking at this graph, having a tough time telling apart this part from that part. For me, they look fine. Your mileage may vary. Whoever made this figure, not good. Don't ever use green and red to show opposing things in a graph. Blue and red are really good contrasting colors. Purple and orange work really well. Yellow is usually light. If it's more of a gold yellow like this, then you're fine. A lot of times yellow is too light to see, especially if the projector isn't great. But red and blue always work really well. Purple and orange are good. Orange and blue are fine. I don't like that because I grew up a Yankees fan, so I don't like to use orange and blue, but that's just me. But don't use red and green. Just FYI for everybody, in your future endeavors, anytime you're making anything graphically, don't use red and green together, because that can confuse people. So we get all these signals from one neuron to the next. And we've been talking about this in the context of just one synapse. What is that one synapse going to do? But in real life, each neuron does not have one synapse attached to it that it can receive signals from. In real life, each neuron looks like this, where you have your postsynaptic cell right here. This is the cell body of the postsynaptic neuron. And then each of these things are a synaptic terminal signaling it. So every neuron is going to have dozens or hundreds or even thousands of axons targeting it. And it's not going to receive one signal at a time. It's going to receive dozens or hundreds or thousands of signals simultaneously. Some of those signals are going to be excitatory, some of those signals are going to be inhibitory. So how do we know what happens in that postsynaptic cell? Well, that cell has to take all of those signals and basically figure out what the net change in membrane potential is. And we call that summation. And there's a bunch of different factors that go into it. But basically, if you have an inhibitory signal over here, an excitatory signal over there, another excitatory signal over here, what's the net change? If that one's minus 10 and this one's plus 5 and that one's plus 10, then what's our net change in membrane potential? And do we achieve the threshold? You don't actually have to do that math. It wasn't actually asking you to do the math. It was just think about it conceptually. But I appreciate your initiative. So the idea is that you're never going to get just like one signal at a time, and you're never going to get all excitatory or all inhibitory signals at the same time. You're always going to get a mix. So what you're going to end up with in your kind of net postsynaptic potential, it's going to be due to a combination of factors. Things like the interactions there. Right, your -5 hyperpolarization, your +10 depolarization works out to a net of +5, right? In that simple example. But the distance from the axon also matters, because as we'll see in a minute, this process takes place at the axon hillock, right? That's where you determine, am I going to do an action potential or not? So if you have a synapse that's all the way on the far side of the cell body, from that axon hillock, well, those ions from that, say it's a depolarization, those ions have to diffuse all the way across the cytoplasm. So by the time those ions diffuse, it's going to be a fairly weak signal. It's not going to exert that much influence on what's going on at the axon hillock. At the same time, if you have a synaptic terminal that's like right there next to the axon hillock, that's going to play a much larger role in determining what the situation is for membrane potential at the axon. So the distance to the axon actually matters within that cell. And then the third thing is the concentration of the neurotransmitter. Right? So remember, square brackets means concentration. So if the concentration of the neurotransmitter, a lot of neurotransmitter is going to open up more channels, you're going to get a stronger depolarization or stronger hyperpolarization. Less neurotransmitter gets released, fewer channels get opened, weaker signal. So you have to take all these different factors into account when you're figuring out what's going to be the net effect of all the signals that a neuron is receiving at any given time.
Why is neurotransmitter in the brackets?
That means concentration. That's a habit I picked up from chemistry classes in undergrad and it never went away. Unless I've had to write the word concentration, I don't anymore. It's been that way for longer than I'll say. Any other questions at this point? Okay. All right, so there are two ways we can add up signals in a neuron when we're doing summation. The first is called temporal summation. And the thing I want to emphasize about temporal summation is that it's going to be multiple signals overlapping at the same synapse. So when you get a signal, you're going to have a change in membrane potential, and then that change, if you don't hit the threshold, that change is going to dissipate. Right? So you depolarize, and then it goes back to the baseline. Temporal summation is where you get two or more signals at the same synapse so rapidly that the first one hasn't entirely dissipated when the second one arrives. So the magnitude of those depolarizations is going to stack, they're going to overlap. And this results in a stronger depolarization. Now, why does this matter? It matters because usually one single depolarization, one burst of neurotransmitters leading to one depolarization in the postsynaptic neuron is not enough to reach the threshold potential. That single depolarization isn't going to do it. But if you can have multiple depolarizations occurring in a row, where they overlap, where the second one arrives before the first one dissipates, and then the third one arrives before the second one dissipates, you can stack them on top of each other, and by doing that, you can achieve the threshold potential and trigger an action potential. So we're going to look at this graphically here. In this cell right here, so this is our postsynaptic neuron, and it has a bunch of connections. E means excitatory, so we have two sites where there are excitatory signals can come in, and then I is inhibitory. Now we're only going to focus on E1 right now, just this one right here. So if you have a slow or low frequency of action potentials, you're never going to trigger an action potential in the postsynaptic cell. Because one action potential will arrive, it'll trigger a depolarization in that postsynaptic cell, and that depolarization dissipates. And then you hit it again, but it dissipates. And you can do that as many times as you want in a row. It's never going to hit the threshold. It's never going to trigger an action potential. But if you look right here, now we have two arrows. Now we've got more action potentials arriving very rapidly on top of each other. So now we have the first depolarization, and it starts to dissipate, but then the second one arrives. And instead of starting from your baseline, you start from an already depolarized state, and you depolarize it more. Now you hit the threshold, now you can trigger an action potential. Okay, so that's how temporal summation works. It's a single synapse where you have two or more signals arriving so quickly that their effects on the postsynaptic potential overlap. And that's one way you could potentially achieve an action potential in the postsynaptic cell. Question?
I have two questions. So why does the resting potential start at -70, and what is the dotted line necessary?
So the -70 is just our equilibrium state for our membrane potential, given sodium-potassium pump and the leaky potassium channels and some other stuff we didn't talk about. That's just our baseline state. And then the dotted line is the threshold potential that you need to hit to trigger action potential. So the dotted line is about -55, and that's what we talked about last time. That opens up the voltage-gated sodium channels. So if you don't hit that threshold, you don't open up the voltage-gated channels, no action potential happens. And once you hit it, then you're good. Second, you got that one. Did we get both? You got both.
What does FP stand for?
Membrane potential. MP is membrane potential. Other questions? Okay, let's keep going. The other type of summation is called spatial summation. And this is where two signals, this is actually a little bit imprecise, because it doesn't just have to be excitatory. This is where two signals or more arrive at about the same time from different synapses. So you have one synapse, second synapse, those signals arrive together, and their effects are going to interact. If they are synergistic with each other, right? If they're both excitatory, then they're going to stack. So you can get a stronger depolarization and potentially achieve the threshold potential. If they are antagonistic to each other, if they're opposite, then they might cancel each other out, either in part or in full, depending on the magnitude of that depolarization. I feel like the mic is getting weird. Is it like coming in and out? Yeah? If it breaks, I'm just going to cry and we'll just end class. We'll just be done. I don't want to fight with you today. So we'll see how far we get. Okay, so let's look at graphically again. Let's see what spatial summation looks like. So we have two examples here. The first is where we have a synergistic effect. So we have excitatory 1 and excitatory 2. Those two signals arrive together, so their combined depolarization is the sum of the two of them, which is much stronger, and it allows you to hit the threshold and trigger an action potential. In this example, we have an excitatory signal arriving at the same time as an inhibitory signal. So in this graph right here, you can see we have your depolarization from E1 And then separately, what does the IPSP look like from here? And then the net result of that is shown right here, where the magnitude of depolarization is greater than the magnitude of the hyperpolarization. So when they overlap, the net effect is to have a depolarization, but a weaker one than you would otherwise have, because you're just taking those two independent signals and you're adding them together, and the inhibitory signal partially cancels out the excitatory signal. So that's what can happen with spatial summation when the signals are in opposite directions. They're antagonistic to each other. Is everybody okay with that? Now, why are we taking so much time to talk about spatial and temporal summation? In a little bit, we're going to talk about long-term potentiation. And in order to achieve long-term potentiation, we need both of these. So we need to understand how both spatial and temporal summation work in order to see how we cause the development of long-term potentiation in our neurons. So these interact at the axon hillock. As I said, that's where all of these different signals are integrated together. The axon hillock is where summation occurs. So the membrane potential at the axon hillock represents the combination of all of the different postsynaptic potentials that are being induced at any specific time within a postsynaptic neuron. And any neuron in your nervous system can at any point be a postsynaptic neuron, which is where it's receiving signals from other neurons. So we take all of those signals together, and it's at the axon hillock, remember the very base of the axon, where it starts to extend away from the cell body. That's where we're doing this math, and we're seeing if we hit that threshold potential and generate an action potential. So we have all our signals coming in, like we just saw. This is the same thing we just saw, whether it's temporal summation or spatial summation. The idea is that you're going to get these signals to the dendrites of the cell body, but you won't actually make an action potential, back here in dendrite. You don't make action potentials in dendrites. You start it right here at the axon hillock, and if you're successful, then that's going to transmit down that axon. And again, I think back to last time, it's an all or nothing thing. It's not like, well, you hit the threshold, there's some inhibitory signals, so yeah, it's action potential, but it's weaker. It's not how it works. If you hit the threshold, you do an action potential. If not, you don't, and that's the end. And it starts at the axon hillock. Questions about anything synapses before we continue?
My first question is, you said antagonistic to each other, they might cancel each other out. Are there any cases that they would cancel each other out?
If they're antagonistic, they work against each other. So like if you have an inhibitory signal that depolarizes by 10 millivolts, you have an excitatory signal, that hyperpolarizes by 10 millivolts. You have an excitatory signal that depolarizes by 5. We're just going to add them together, right? The net effect there would be -5. They just cancel in opposite directions.
And then for going into the memory part too, so if there's I don't want to say blockage, but if something were to happen to a synapse, right? Then does that involve memory?
Yeah. So if something messes with your synapses, right, then you can't do this. So you can't, for example, access the memories associated with a specific set. We're going to talk about memory in a minute, but to the extent there's a physical medium for memory, it's synapses. We know that forming new memories is associated with physically forming new synapses. And so if you mess with the process of forming a synapse or the signaling across newly formed synapses, that makes it hard to form new memories or recall those memories when you need them. Yeah. Question? Yeah.
So and there's nothing at all that can alter the actual action potential. Just getting there.
Right. Right. Once you hit the threshold, you're off to the races, right? And you've done it. You've triggered the voltage-gated channels. Now, in theory, I'm sure there's an obscure neurological condition where there's a problem with sodium voltage-gated channels. Then the action potentials don't work as well as they should, right? I'm sure that's a thing. But for our purposes today, where once you hit that threshold, you do the thing. And that curve is going to look the same way, assuming everything is working properly. So let's talk about memory and learning, because synapses is not just about moving your muscles the way you want to move them. Synapses are also like the thing when it comes to forming memories. It's all about changes to your synapses. So we're going to take just a couple of minutes to review a little bit. There's nothing new on this slide. This is all just stuff that we talked about last semester and also previously this semester, but it's just to kind of frame where we're going here. So if you think about your nervous system organization, the macro scale, the large scale organization of your nervous system is established during development. We talked about this just a few days ago. We talked about the process of neurulation and then the structure of the nervous system coming out of that. Right? Your neural tube is going to form your central nervous system, your dorsal hollow nerve cord, and your brain. We talked about the parts of the brain. And then radiating from that, you get your peripheral nerves, your sensory neurons, all that stuff. But that's just the overall pattern, right? That's not the specifics. All of this is regulated. We talked last semester about gene expression. We talked this semester about signal transduction. We talked about induction during neurulation. But then after birth, we're not done, right? Your brain is constantly changing and remodeling. It's extremely dynamic. And it's a little bit counterintuitive because we've talked, I'm sure most if not all of you know, that your central nervous system doesn't regenerate, right? So you don't grow new neurons over the course of your life. But it's still a highly dynamic system, and that's all about the connections between the neurons that are there. It's all about constantly forming new connections, getting rid of old ones that you're not using, just remodeling the actual circuitry of those connections in your brain. So this concept is called neuronal plasticity, which is a term that we introduced in the first class of the semester all the way back in September. We're going to go into this in a little bit more detail, and then we'll get into types of memory and long-term potentiation in a bit more detail. Doing great on time. So neuronal plasticity is all about remodeling, and it really is driven by competition for things that your neurons need to function. So you have these things called growth supporting factors. We don't need to talk about what they are, but basically these are chemical signals that facilitate the development of your neurons. So forming new synapses, strengthening those synapses. And the competition is kind of analogous to natural selection, but instead of competing for resources, or what I should say is that instead of the success in the competition being based on fitness, success here is measured by activity. The neurons and the synapses that you use, they're the ones that win the competition. The ones that you don't use are the ones that lose the competition. What that means in practice is that for synapses that you use, you're going to get more of them. And for synapses that you don't use, they're actually going to be eliminated. So we can look at this figure. We have up here, we have 1, and then down here we have 2. Now connection number 1, synapse number 1 right here, you see those little red arrows. That means action potential, so we're using this one a lot. In the before picture, we have 1 synapse connecting these 2 cells here. But after a period of use, over some amount of time, now we have 3. So we've increased the strength of that connection through use. And then down here, the opposite is happening. We have a connection, but we're not using it. And these connections are really expensive to maintain. Just think about the cost of constantly synthesizing neurotransmitters and synaptic vesicles. They don't last forever, right? They wear out over time, even if you're not using that synapse. If you're constantly making more of them, that's really energetically demanding. Making the receptors, the postsynaptic cell, really energetically demanding. Setting up all those gradients and using all the gradients, really energetically demanding. So if you have a synapse right there, and you're not using it, that's a waste. So get rid of it. You can put that energy towards something we're actually using. So this use it or lose it dynamic underlies the constant remodeling of your central nervous system. And this is the fundamental thing that underlies memory formation and learning. In addition to the actual presence or absence of synapses, we can zoom in and we can actually look at the strength of those synapses. And so the connections themselves can be modified, and this often happens when you have two or more synapses that are operating together over some period of time. So in this example, we have 1 and 2, they're firing together. They are going to get stronger as a result of that mutual reinforcement. This is why associative learning is so powerful. This is why you can form a connection between two different things in your brain, and then later on use that connection to help you remember one of them. You can trigger one, and that allows you to get the other one. And it's because you trigger the memory associated with this one, it's going to make it easier for this neuron to operate, because they operate together. Okay, so they're active together, that strengthens both. The saying that somebody told me one time was, it's very right, it was neurons that wire together, fire together. So if you can have two synapses operating simultaneously, you strengthen those connections together through associative learning, then you trigger one of them, and it makes it easier to trigger the other one. And you could really exploit this. There's a lot of like little tricks you can do to exploit this, to help you remember things. So what I would do when I was an undergraduate, again, I won't say how long ago that was, but to give you an idea, it was before iPhones existed. So what I would do is I would make, don't make that face. I would make like a playlist for myself. Every time I would study for an exam, it would be a different set of songs for each exam. And I'd put it on my iPod with the little wheel thing and my wired earbuds, and I would listen.
iPod Shuffle?
No, it was before that. It was like with the little screen, yeah, the little black and white screen. It was like a Game Boy, but you know, it was just for music. But this existed. It was a while ago. So I'd make this playlist. It would only be short playlists, 45 minutes, an hour, and I listened to the same set of songs the whole time I was studying for the exam. Then I'm going to walk across campus to the exam, listen to that playlist on my walk across campus, and then I have those songs going in my head kind of during the exam, keeping me company, and that helped me remember the information for the exam. There's all sorts of little tricks you can do like that. One thing you can do if you take your exams in this room, sit in the same seat for the exam as you do in class. Because just like the orientation of your body when you're thinking about the material will help you remember the material. That's just like a weird little thing. You can chew a certain flavor of gum while you're studying and then chew that same gum during the exam. Weird little things like that. You can associate memories that help you remember things. There's an underlying neurological reason why little tricks like that work, and they are totally legitimate. Use it to your advantage. Big idea. Use it or lose it, right? You form memories through prolonged stimulation and use and practice. And that makes it both easier to remember the thing, like have it in your long-term memory, and it also makes it easier to use it when you need it. If you don't do something for some amount of time, like using an example just at random for no reason, let's say you have a final exam coming up in like 4 weeks, and there's stuff from a couple of months ago that you haven't looked at since you had an exam on it, hypothetically. Are you going to have an easy time recalling that stuff on the final exam if you're right? So just hypothetically, if that's a situation you might find yourself in at some point, it might be useful to revisit that older stuff so that you're able to recall it when you need it on that hypothetical final exam that's coming up in 4 weeks that is cumulative. So let's talk about memory and learning. There are two types of memory there's three, but one of them is really short, we don't care about it. The two types that we care about, the first one is short-term memory, which is where information is held for a very short time in your brain and then it is released. We can talk about this in a little more detail than we did way back in September, because we've now talked about like the structure of brains. So I can tell you, for example, that short-term memories are stored in the cerebral cortex, but you don't form new synapses for short-term memories. You're using pre-existing connections via the hippocampus. And I always analogize this to RAM, right? It's your RAM in your computer, and the more RAM you have, the more stuff you can do at a time, right? And maybe you have 8 gigs of RAM, like that's pretty good. but then you have 50 tabs open on Chrome, and you're like trying to render, some video in 4K, and like all of a sudden your computer gets really slow and bogged down because there's just not enough of those temporary connections in your RAM to cover it all. That's how your short-term memory works. So you can store a few things in there, but if you try to add more to it, the connections are temporary, so you're just going to pump out the things you're not using at any given time. So this is really important for acquiring memories. The first step in forming your long-term memories is getting them into your short-term memory, but it's not going to be maintained. So the last thing on here is capacity and duration. So what's the duration for short-term memory? Give it like approximately. Yeah, like seconds to minutes, right? Not longer than that. And what's the capacity? What did they tell you in psychology class? There's like a bunch of different ways to say it. What did they say in psych? 5 to 7, 5 to 7, about 7, 7 plus or minus 2, right? So it heard something like that. So I always just say about 7, right? So it's limited in time, seconds to minutes, and limited in quantity to approximately 7 items. This also is useful information for you in a practical sense. Because who has ever come to an exam room half an hour early, sit down, you're flipping through your notes, or you're reading the textbook? Yeah, I don't do that. I never did that either. But just, and if you've done that, you spend 20 minutes reading through your notes, what's the only thing you're going to remember?
The last thing you see.
The last page of your notes. Because all that stuff you're sitting there reading is in your short-term memory. So let's say for this exam we have coming up, it spans from plant physiology to nervous system 1, right? The very last topic, sorry, I didn't mean to scare you. The very last topic is brain structure. So if you're going through all your notes, right, and you finish and you're going through the different parts of the brain, and then we start handing out the exam, and a couple minutes later we start the exam, what should you do? Should you start with the plant stuff? Now, you're going to read 2 questions, all that brain stuff is gone, right? What you should do is flip to the end of the exam and find the questions about the parts of the brain and answer those real fast while that stuff is in your short-term memory. So as soon as you read that first question about xylem and phloem, all that stuff about the parts of the brain is gone. Because that was in your short-term memory, and now the first question about xylem and phloem is in your short-term memory. All right, so if you're going to do that, do whatever works for you on the exam. If it makes you comfortable to get to the room half an hour early, sit there reading your notes before the exam, 100% go for it. But then do the next step in the productive way and make sure that the first questions you answer on the exam are on the last topic you read before the exam started. That's taking advantage of your short-term memory. You all see how I'm trying to help you out here today? I'm trying to all right. The other type of memory is long-term memory. So this is where information is stored in the cerebral cortex. It's not involving the hippocampus anymore because we are making new long-term connections. We're forming new synapses. And again, to the extent there is a physical medium for memory, it's new synapses. Now, in terms of duration and capacity, long-term memory is theoretically permanent. In some cases that, seems to be the case. People remember stuff from their childhood, very late in life. Is it truly permanent? There's definitely still some use it or lose it to it, but permanent-ish. And then capacity-wise, as theoretically unlimited capacity, and experimentally, we haven't been able to find a limit for the capacity. In theory, there is a limit for the capacity because your brain has a finite number of cells. There's only so much energy in your body that you can put towards making synapses. But if there is an upper limit, we haven't found it in a practical sense. So what we can say is that the duration and the capacity for long-term memory are both theoretically unlimited. In practical terms, there's certain limits, but we haven't been able to concretely identify. Is everybody okay with that? Well, so theoretically unlimited. So that brings us to a super important topic, long-term potentiation. Some of you may recognize this flowchart from the very beginning of last semester if you encoded it successfully into your long-term memory. If you didn't, here it is again. This is another exposure, and hopefully with repeated exposure, you can encode it into your long-term memory. So what we see here is the basic process of memory formation. You have your sensory memory storage, which is like the milliseconds one, that's just every sensory thing that comes in goes there. If you consciously kind of detect something, if you consciously notice something, then even for just a brief moment, it's going to go into your short-term memory. And most of that stuff just gets forgotten. But if you work with something repeatedly, it's going to get encoded into your long-term memory, and that's where you want it. That's memories that you have stored, you can access those. But the trick is that you can't use your long-term memory directly. You have to retrieve it into your short-term memory in order to use it. This is again why I like the RAM analogy. This is like your hard drive, although with solid-state drives, the analogy doesn't really work anymore, but pretend, old spinning disk hard drive is your long-term memory. This process of going back and forth between short-term and long-term memory, that's the critical process for us, because that is what induces long-term potentiation. Now, yesterday, somebody asked a really good question, because we just talked about the duration for short-term and long-term memory. They said, okay, we talked about the duration. How long does it take to do this? How long does it take to actually get something into your long-term memory and then facilitate or form long-term potentiation? And the answer is that it takes anywhere from a few days to a few weeks or longer, depending on how you do it. But what you need to do is you need to have repeated use or exposure or practice or whatever verb you want to use with related stimuli. You'll see why it has to be more than one thing. There has to be related stimuli, more than one thing, together, over a period of time, days or weeks at a minimum, with sleep in between. Those are the three things you need. And yesterday, that led to a lot of very panicked faces in class, because people were going, oh, that's not how I do it, right? And there's a good reason for why all those things have to be the case they are. And we're going to go through in a little more detail and talk about how we actually physically form long-term potentiation. We're going to see why it has to be multiple stimuli over a period of time. And then sleeping is important, because sleeping is when your body's not doing much else, so that's when your brain can really take stock and form new synapses and things. It's also when your cerebrospinal fluid gets to kind of circulate a little bit more. So sleeping is a big important part of it. So there are two conditions that we need to meet at kind of the molecular level. We're not talking macro level, we're talking the molecular level for long-term potentiation, which, by the way, I don't even think I defined it today, but long-term potentiation is a lasting increase in the strength of synaptic transmission. So you are better able to transmit the signal across a synapse if that synapse has undergone long-term potentiation. So there's two things you need for this to work kind of at a micro level. You need a high-frequency series of action potentials. In other words, you need temporal summation. And you need depolarization from another stimulus at the same time, which means you need spatial summation. So we're going to see why each of these things is the case at the molecular level. We're going to zoom into that synapse and see what changes occur when we induce long-term potentiation. Now the idea here is we're going to talk about one specific synapse that's associated with one specific memory, right? And the point of long-term potentiation is to be able to retrieve that memory when you need it without any help, without another stimulus kind of helping you along. So an example of what you may have experienced at some point in your life when you don't have long-term potentiation is that you have something in your memory and you know it's there. Like, you read a question on an exam. You've probably experienced this. I know I have. You read a question on an exam, and you know the answer. And you finish the whole exam, and you go back to that one question, and you're like, I know it's right there. I know the answer to that question. And you just can't quite get it out. And then after the exam, you go to your notes, you go to the textbook, you open up the book, you read the first two words on page 136 that's relevant to the thing, and now you can just write a whole paragraph on that question. That you could, a bunch of people yep, been there. What happened is the synapse, that pathway that was associated with that information, getting it from your long-term memory to your short-term memory, it didn't have long-term potentiation, so you couldn't, at one step in that chain, you couldn't trigger an action potential across that synapse. You couldn't hit the threshold in the postsynaptic cell. When you read the book after the exam, that was a depolarization of the same cell because it's related information. So that was a second stimulus for the same pathway, and combined, that spatial summation allowed you to trigger that action potential, and you could recall the information that you needed. That's what happened there. The goal with long-term potentiation is to be able to trigger the action potentials through that pathway without needing the spatial summation. And that's the goal. We want to make it so that you don't need to have that other prompt to get over the threshold potential. So if you have these two things, we're going to go through the process. It's going to start with a lot of spatial summation. That's your practice. And then when you're recalling the information, when you achieve long-term potentiation, you're going to have temporal summation doing the job. What's going to happen as you do this is your synapses that you associate together, right? How long-term potentiation works, and the flavor of gum I was chewing in class while I took notes on it, and also while I practiced it, right? You associate those two things together, it's going to strengthen both of them. So that one of them is going to help you recall the other one, and it works in both ways, by the way. The gum will help you remember the information, but also then for the rest of your life, anytime you think about synapses, you'll then think about like spearmint gum or whatever it is, right? It works both ways. But the other effect of it is that you can then recall that information when you need it without needing that other stimulus to help. Right? That's what's going to happen. So let's get into the details. We're going to do this in words first, and then I'm going to show you the pictures. When we get to the pictures, just so you're clear, there's going to be a lot of detail on there, like the names of specific receptors, the names of the neurotransmitter. You don't need to know any of those specifics. It's the principle of what's going on step by step.
So about spatial summation, like an example, like what if you listen to one song anytime you study?
Yeah.
Would, like the advantage of spatial summation be like reduced, or would it like?
If it's like one song
for like whatever.
for all of bio, that might make it harder because then that song opens up a bunch of different things that are not necessarily connected to each other, right? That was why, this was kind of trial and error for me. Again, I'm just describing my experience. Your mileage may vary. But that 45 minutes to an hour was good because the exam, it's like an 80-minute period, or I forget on the crowd. I think it was an 80-minute period. And so, I have those songs kind of going in my head, and I kind of run through all of them once or twice over the course of that exam period. And each one would be a different kind of thing that would kind of run through, kind of helping myself remember the stuff. But if it's one thing, then if it's one 4-minute song, one 3-minute song over and over again, that's associated with everything. I would associate it with not one thing, but all the things. That might confuse you more, I would think. But again, your mileage may vary. Do whatever works for you. Or you could split the difference. You could do one song that's 45, you do like Beethoven's Sixth Symphony or something. It's like 50 minutes long, and there you're good. Yeah. Okay, so what I need you to understand, again, not specific types of receptors, but hang on one second. Let's try that. All right, not specific types of receptors, but just the idea that there are different types of receptors. There are going to be 2 types of ligand-gated ion channels in our neurons at the postsynaptic membrane. One of them is always in the membrane, and the other one is not. It's just kind of hanging out in storage, right below the membrane, but it's not actually in the membrane. It's not actually a transmembrane protein at that point. So it has no extracellular component to it. It can't detect any neurotransmitters. This means that there just aren't that many receptors in the membrane before you establish long-term potentiation. So if you want to depolarize that postsynaptic cell enough to trigger an action potential, you need to hit it from more than one place at a time. You need to do spatial summation, because any specific synapse on its own is not efficient enough to trigger a strong enough depolarization. Right? That's your kind of before state. Now, after long-term potentiation, you're going to have both of those types of receptors in the membrane at the synapse that you strengthened. So you no longer need a signal from another synapse to help you depolarize. You have enough receptors right there. The only thing you need after long-term potentiation is temporal summation. You hit multiple signals at that same synapse, they're going to stack on each other, and they're going to be strong enough to achieve the threshold potential and trigger an action potential in the postsynaptic cell. Right? That's the point of long-term potentiation. You strengthen a synapse by physically adding more receptors to it, and those ligand-gated receptors allow you to trigger a stronger depolarization from a single synapse. Conceptually, is everybody okay with that? We're going to look at it graphically here, but conceptually, how do we feel about that? We start with a very inefficient synapse that requires spatial summation to work. Over time, you build up that strength. Now it becomes a very efficient synapse that doesn't need any help from elsewhere. It can operate all on its own. That's the idea. So we're going to look at this in picture form. There are, again, pictures are more detailed than you need to know, so don't worry about the specific types of receptors or anything. But I just want to show you kind of visually how these changes actually look at the molecular level. So here's our before state. We have two types of receptors, purple in the membrane, pink below the membrane, in storage. Now the purple receptors, in addition to them not being, there's not that many, right, because a bunch of receptors are stored down here. In addition to not having that many, they also get blocked by magnesium. So even if you stimulate those receptors with a neurotransmitter, they don't work that well. Right? So you have a weak depolarization, if anything at all, before long-term potentiation. You'll notice there are specific names of receptors and glutamate. We don't care about any of that stuff. Just follow me conceptually. That's what I want you to get out of this. So now, when we're in the process of establishing long-term potentiation, so now you're practicing, you're workshop, you're drawing stuff, you're looking at your notes, you're drawing things, you're talking to people, you're presenting and answering questions, right? These are all different stimuli using the same pathways. So we're all going to be mutually reinforcing. So what's happening here, if we can continue this postsynaptic membrane, there's going to be another synapse, like over here somewhere. You're going to depolarize that synapse at the same time. Those ions from over here, they're going to diffuse throughout that cell, right? They don't just stay in that one place. They're going to diffuse. That's going to lead to a depolarization over here. And a depolarization is what unblocks those original receptors, that boots the magnesium out of it. So now those receptors become more efficient every time you depolarize. While that cell is already depolarized via spatial summation, the depolarization from this particular synapse gets stronger. But then, of course, the spatial summation goes away, and that goes away. But over time, you actually make a change to this membrane. You add those other receptors. Hitting this again and again tells your brain, hey, let's make sure that synapse can work well. Let's make sure that works when you need it to, because we're using it all the time. So let's make sure it really works. So you add the pink receptors to that membrane. Now, after days, weeks of practice and associating those different signals and stimuli together, now we've established long-term potentiation. So now we don't need spatial summation anymore. Now we just need temporal summation to trigger an action potential here. Because that first signal that comes through, it's only going to have the pink receptors available. The purple ones get blocked by magnesium, so we can't use those initially. But how do we unblock them? We unblock them with a depolarization. The pink ones provide the depolarization to unblock those other receptors. So now the second action potential that comes down here, the second burst of neurotransmitters, you can use all the receptors in that membrane, the pink and the purple, which means you get a much stronger depolarization. Maybe the second one doesn't get you to the threshold, but the third, the fourth, the fifth, as they keep stacking on top of each other, you won't need help from somewhere else. You won't need another stimulus and another synapse to help you get over that hump. You're going to be able to depolarize that cell from this one synapse enough to trigger an action potential in the postsynaptic cell. Right? That's the point of long-term potentiation. that you change the biochemical arrangement, you change the biochemical makeup of that synapse to where you can trigger an action potential from a single synapse without needing help from other connections targeting that same cell. And what this means in practical terms is you can read the question on the exam and you can recall that information into your short-term memory when you need it without needing the prompt of just seeing the first few words of the sentence in your textbook. Right? That's what this accomplishes. And again, have I said the names of any specific receptors? No. It's all about the different types of receptors and what gets you to add them to the membrane and the effects of that happening. Questions?
So is it like purple slump magnesium? So there's lots of potential, and then it's depolarized in different kinds of slump. So this is magnesium, and then to like double check, I guess, the pink is added, and sodium sodium, and it depolarizes blocks purple, and triggers.
It unblocks the purple. Yeah. So in your starting state, you can even think about it simpler than you said it. In your baseline state, you only have purple receptors, and they're very inefficient. So in order to even depolarize this synapse right here, you need spatial summation from somewhere else to even just like jumpstart that process. It can't do it on its own. But with prolonged periods of association between this synapse and one or more others in that same postsynaptic neuron, you're not only going to allow those purple receptors to work every time you depolarize it, because that unblocks them, but you're also going to tell that cell to add the pink receptors. Once you have the pink receptors there, now you don't need help anymore, because the pink receptors can do the initial depolarization, and then any subsequent hits, so your temporal summation, right, a bunch of signals really fast, that's going to open up all the receptors, and you could hit the threshold without any outside. Good? Great job. Yeah, wait. It's so these are specific receptors. There's all different types of receptors we're not talking about, but this basic process is what you need for long-term potentiation, where you're adding receptors to the membrane that allows you to do temporal summation rather than requiring spatial summation. But you need to do the spatial summation to induce that change. Yeah, so the repeated depolarization of this cell is what triggers the use of the second type of receptors. Basically, if you depolarize a lot, you say, hey, use this a lot, let's make it work better, please. Right, that's the idea. Okay, so let's settle a little bit here, right? This is the point of workshop, right? This is what we do. You're listening to me go on for 80 minutes at a time, you're reading your book, you're reading your notes. Those things are single kind of independent stimuli. Those things are not going to trigger long-term potentiation. But if you do the things that we try to get you to practice inside and outside of workshop, sequence, compare and contrast, discuss things with each other, come up with system-level questions and quiz each other, those things involve more than one stimuli. So what you're doing is spatial summation for the pathways involved in those memories that you're forming for the course content. When you do that for a prolonged period of time, specifically using these words like sequence, compare, contrast, hypothesize, identify, all those words that we use, when you see on the exam, sequence, blah, 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 that stimulates the pathway in the brain, and if you achieve long-term potentiation, guess what? You don't need any help. You can just pull that information when you need it, because that one pathway is going to allow you to retrieve it into short-term memory. Right? So it's all about telling your brain, this is important, keep it, and be able to access it when I need it. That's the signal you're sending your brain by practicing with this material in an active way over a period of time. There's a simple way to kind of test yourself to see if the things you're doing to study are working. And it's this. If you finish studying, like you're sitting down at your desk or whatever, you're doing something for an hour or two, and at the end of that hour or two, you're like, I feel fine, I'm going to keep reading the textbook. That was great. I'm just going to read for another hour. If you're not tired, it didn't work. Because, like, these processes that we're talking about involve using energy. Right? It takes a lot of energy to change the arrangement of your synapses and use lots of neurotransmitters, like this little energetically demanding stuff. If you don't feel tired after doing it, like physically, then you're not accomplishing much. You should feel tired after studying, or I prefer the word practicing, right? After practicing, you should feel a little bit tired because it uses a lot of energy if you're doing it right. If you're just going through like flashcards or something, you know, made yourself like a Quizlet, it's just flashcards with definitions, you can sit there and flip through those all day long, and you feel fine, then you're not forming any new synapses because that's an energetically demanding thing to do. And you'll get tired doing it, but that's how you know it's working, right? So if you get tired after practicing, that's great, take a break, then go back to it, you know, the next day, later day, whatever, right? But if you don't feel tired afterwards, then change the way you're approaching it, because you're not actually accomplishing these things. Because these are energetically demanding things, and your body doesn't care if you're using ATP and your muscles are in your brain. It's still using ATP, it's still costing you energy, it's still going to make you tired. Oh, sorry, I thought that was a hand up. Good. Okay, so let's summarize synapses. It's connections between neurons. We talked about electrical or chemical synapses, but we're all about chemical, whatever. As we talked about, there's lots of different inputs that are all going to get summed to get a specific effect. And we talked about long-term potentiation and how that affects learning. Now, obviously, we are not in 2 minutes going to cover sensory receptors today, which is good. We get to stop at exactly the same time as our other class. So we will pick up next time talking about sensory reception and sensory processes, and we'll talk about eyes. The practice question here is about vision, so I won't show you that. That would be pointless today. So I will just show you what we're going to do next time. After sensory receptors, we're going to talk about the musculoskeletal system, and it's really fun. So that's what we're going to do next time after we talk about sensory reception and eyes. So thank you, everybody. Remember, we have a review upstairs in room 205 immediately after this class. Typically, it is nice and warm in that room as long as the windows aren't open. So come on up and get warm and talk about bio some more, because that's what we all want to do on Fridays, more bio. Thanks, everybody. Have a great weekend. See you on Tuesday.